One of my students, she's right now in Caltech in California, unfortunately very far away. But she's been doing a great review, and she's been doing a lot of great study on biological aging clocks, and she will present briefly her work on biological aging clocks, methylation clocks, and epigenetic clocks.
Wow, thanks for hearing today. It's incredible. These three amazing women are paving our path for the future, and I just really want to acknowledge their work. Evelyne, Andrea, Stevie, thank you so much. Let's give them a round of applause. And Evelyne, thank you so much for giving me the chance to speak today. We've been working on a very interesting project with Evelyne. It's the use of epigenetic clocks in clinics. So today we can measure blood, the liver, the heart, lungs, everything. We can give each one of them an age, a biological age. And it's wonderful the work that's been going on so far. It started in 2011 with Blockland creating the first epigenetic aging clock. But so far there's been lots and lots of research, but little application of these clocks in the clinic, which is what we would want to emphasize today. So the reason why clinics would benefit so much from these clocks is that they not only give the biological age, but they can also show the pace of aging, so that people can track how well treatments are working, and therefore lay out treatment trajectories for their patients. Now what we did is that we collected through all existing literature all of the epigenetic aging clocks that currently exist. And epigenetic aging clocks, they differ from other biological aging clocks. These are the most well-studied clocks. They have a lot of evidence for them. They're based on CPG sites, which show the expression of certain genes, and you can measure this expression and compare it between different people. And what we did was we analyzed and really dug deep into each one of these clocks in order to understand which one is the best, which one has good characteristics, and which one is useful for an actual clinical setting. And reviewed them according to strict criteria, and from all this research determined what the criteria of a perfect, a dream epigenetic aging clock would be. So here's a short diagram, a small diagram of what we've been looking at, and it shows you the most fundamental epigenetic aging clocks currently in the field and what's been going on. So you can see a lot. The ones that are the earliest have the most simple features, but they lay really the base and the foundation, whereas the later ones are based on deep learning and artificial intelligence and many more complex algorithms. And this is the criteria by which we analyze each of these clocks. So we had to make sure that the method of making these clocks is reliable. We looked at the number of CPG sites that each clock used, and we also considered, for example, the sample number. How much data did people actually have when making these clocks? We ensured that there were different tissues studied, for example, blood, saliva, and we also looked at the applicability to different cell types, so whether people in the clinic will be able to use it just for one tissue type or not, or others. Of course, it's important to make sure that these clocks are good for males and females, and as well for different ethnic origins too. And we also evaluated whether these clocks were looking at disease-related effects on aging, among other factors. From there, we devised what the dream epigenetic aging clock would be. And these are the different criteria that we came up with. First of all, it has to be easy to implement and easy to use in the clinical setting, otherwise it won't be accepted as well. It has to require a minimal number of samples in order to actually function with high accuracy. What's important is that it predicts mortality, so how much a person, for example, has left to live based on this clock. This is important. It's one of the most fundamental measures for aging. Of course, it has to evaluate things like psychological age, that's related to brain aging. And we want the results to be delivered rapidly. We determined that there is a trade-off between three different factors. So first of all, the accuracy. So how well can we actually, how proper this clock is and how accurate its results are, how much can we believe in. Then there's feasibility. How easily can we actually make this clock and implement it? And finally, predictability. So can the clock predict things like mortality? And the next steps are now to evaluate non-epigenetic aging clocks. There are so many different ones that we can look at and consider for the clinic. finally designing this dream clock. Thank you.
Great work all the year long, despite being a Caltech student. It's just pretty amazing. Thank you so much.